Okay, everyone. Hello. I think I was just talking, but I realized that my microphone was muted. I tried to unmute it, but anyway, welcome everyone. So I think that we can go ahead and get rolling here. I hope all of you are doing well. Currently, I'm not sharing anything with you yet. I'm not even sharing a video, just an audio of myself. But I'm going to go ahead and share my screen with you here in a little bit. But this is one of those strange things. I've never done a review quite like this. So I always feel strange just talking to my screen. But anyway, so I miss all of you. I really do. I miss our interactions in class, but I trust that you are all doing well and are getting ready for this exam on Monday. So a couple of things about the exam. The exam is going to have essentially the same format that the other exams that you took had. It's going to have the 2 matching questions and then the 10 multiple true-false questions and then the 1 short answer question. Of course, it's going to be presented on Canvas, and I made significant announcements about how to access the exam through ProctorU. So in just a second, I'm trying to admit students here. There we go. So make sure that you read those announcements. I've made 2 or 3 of them now on Canvas. There's an extra credit opportunity that's available to all of you if you just attempt to prepare your system and demonstrate that you've prepared your system for ProctorU on Canvas. Okay, so do that extra credit. It's worth 5 points. You might as well do it. So I know that, ProctorU is not the greatest solution in the world. Some of you have misgivings about it and its intrusive nature. Unfortunately, I don't have a lot of options, especially for a class of 220 people. So I do apologize in advance for that. We're having to ask students to do things that we normally wouldn't ask them to do. So anyway, so I'm going to do this. I am recording this session, so I will post the recording of this and a link to it on Canvas later, hopefully this afternoon. So if you have really drastic lag time in the screen sharing that I'm giving you and it's just not working for you, that lag time won't be present in the recording. So keep that in mind as we move forward, okay? I'm going to go ahead and share my screen now with you, and I'm going to also activate a poll. Great. So I'm already getting questions on this. So we're going to run questions through this format, because then you don't all have to type them in the chat of Microsoft Teams, and then I'm trying to keep track of those. It's really hard to do. On this poll everywhere question, you can just vote for them. So go ahead and take out your phones or take out a computer or whatever, and you can start to respond to this and type questions into this, okay? And I'll start to address those. I'm also going to use, because I'm using my Surface, I'm going to go ahead and use this Surface and a whiteboard and essentially write out, any diagram that I think I need to write out. Okay. All right. So let me go ahead and start addressing some of these questions. Go ahead and start populating this with questions. But let me first of all, it says, is the test ProctorU Auto? Yes, it is ProctorU Auto. So that's essentially what the software is, okay? And again, if you missed it when I first started talking, make sure that you look at the announcement page on BC351's homepage. I made 3 announcements in a row about the exam, how to prepare your system for the ability to get 5 extra credit points for that, and that kind of thing, okay? Okay, so let me go ahead and start addressing questions now. So we'll take the top one right now, which is, it just changed, but I'll go ahead and do the one I was just thinking about. Well, how about, because more people are doing the top one, so how specific do we have to know for the pathways, or what specifics of the pathways do we need to know? Because there's a lot of them, as you've noticed. So this is a good question. And by the way, I'm at home, as all of you are. So if you hear my kids in the background, I told them that I'm doing this, but if you hear them, just go ahead and chuckle. You might even hear some dogs bark and that kind of stuff. But anyway, so there's a lot of information on this exam, in particular pathways, and I never discourage memorization of a pathway for a student. Now, here's what I will say with that. Memorizing the pathways alone is not going to help you out, okay? The memorization of them would be helpful just in terms of understanding the language of the exam, okay? So most questions are more conceptual than they are straight, like true or false. The glycolytic pathway has 10 steps in it, which is of course a true statement, but that kind of question is not going to be asked on the exam. More of the questions are going to be conceptual. How to, for instance, how do the pathways connect to one another, okay? So when you look at glycolysis and gluconeogenesis, I would want you to be able to compare and contrast those, the specific details of the fact that they share 7 different enzymes with each other, but they have these unique ones, okay? And those are the irreversible steps. We talk about 4 unique steps in gluconeogenesis and 3 in glycolysis. I'd expect you to know the names of those enzymes and a basic idea of what they are doing and how they are accomplishing those things. Gluconeogenesis takes place in several compartments in the cell, whereas glycolysis only takes place in the cytosol, right? Glycolysis needs a source of NAD+, whereas gluconeogenesis needs a source of NADH. What are the sources of those NAD+? What are the sources of those NADH? Those kind of questions are going to be more of the types of questions that you're going to see on the exam. Now, I will again ask that you look at the learning objectives for this exam because when I again wrote the exam, I went right down the list and essentially wrote questions that address those learning objectives. So that will hopefully give you a fairly good understanding of the details that you would want to know for the pathways. Okay. All right, show the dogs. The dogs are outside right now. So if I hear them come in, I'll call them down and I'll put them up for you. Okay, so but they're outside right now, running around, trouncing around in the snow. So okay, all right. So can you explain how to tell if a redox reaction is endergonic or exergonic? Absolutely. So, the key with this endergonic and exergonic of a redox reaction is going to be based off of the reduction potentials of the reaction. Okay, so if you look at it this way, let me go ahead and draw this out. We have this value here. Oops, I got to get my pen to draw here. We have delta E that is equal to the reduction potential of the acceptor. Just a second. I think that I might have. Here we go. There we go. Let people in. I hear these beeping, and that means I have to let someone into the meeting. So, okay, coming back to this, let's go here. So we have the reduction potential of the acceptor minus the reduction potential of the donor. Okay, and the reduction potentials are essentially a measure of the affinity of a particular molecule or atom for electrons, okay? And you can think about it very simply this way. If a redox reaction is occurring where the acceptor has a higher affinity for electrons than the donor, then that's going to lead to a exergonic redox reaction. Okay, so and of course that means this. Okay, so with our redox acceptor, let's just say that this happens to be 1, whatever our molecule is. Okay, that would be the value of the reduction potential of the acceptor, and let's just say then the value of the electron donor happens to be 0.5. Okay, these different values and the magnitude of them, okay, again, indicate affinity for electron. The acceptor in this case would be to use maybe another term, a stronger magnet for electrons than the donor. Okay, so that acceptor is going to be capable of taking electrons off of the donor. Okay, because it has a higher affinity for the donor, and you'll notice then, for higher affinity for electrons than the donor. Okay, and so what you'll notice here then is that given this very simple equation, right, the change in the reduction potential is going to be 0.5, a positive change. Right now, that then we can look at this equation, delta G is equal to negative NF delta E. Okay, N is the number of electrons being moved. In the redox reaction, right? So in most biological scenarios, we're either looking at 1 or 2 electrons being moved. Okay, many of the reactions that we talk about will be 2 electrons. Okay, so that could be a 2. Right, F is Faraday's constant. Okay, it's a constant. If you really want to know the value for it, it's 96.5 kilojoules per mole per volt. I certainly wouldn't hold you accountable for that. Okay, but that's the value of it. And then finally, the last thing here would be what we just talked about. Okay, and in this case, the scenario that I indicated, we have a positive change in the reduction potential, and therefore that's going to lead to, based off of this right here, the negative sign at the beginning of this equation is going to lead to an exergonic reaction. Okay, now conceptually, hopefully you understood the concept of this and why it would make sense. Again, you have a stronger an acceptor that has a stronger affinity for electrons than the donor. Okay, if I were to switch these, right, move those one to another, so that now my acceptor is 0.5 and my donor is 1.0 in terms of their reduction potential. Well, now I have an acceptor that has a weaker affinity for electrons. And in that case, then that's going to lead to a negative 0.5. Put that right here. Okay, we'll multiply that by the negative sign in the front of our equation, and now we get a positive, an inorganic process. Okay, and of course this should make sense again conceptually, because now my donor is the one with the stronger affinity for electrons, and yet it's having to give up electrons to the acceptor. Okay, and again, you can go back to magnets and think about electrons as metal beads, if you will. The stronger the magnet, the more likely it is that it's going to take the electrons. Okay, all right. Let's go here. Let me have it. I think that. Okay, just make sure that everyone's in. Okay, so let's go back to this here. Okay, now if you also, okay, so I'm getting a lot of dogs. Trust me, if they come inside, I'll try and get them to you. Okay, are you wearing a vest? No, I'm not wearing the vest. So, you know, it's just one of those strange things that I just had to change things up. You know, I mean, here we've been isolated, and so, you know, I haven't worn the vest in a really long time, and I know that brings a certain amount of insecurity to all of you, but don't worry, we're gonna make it through this together. Okay, we can do it with or without the vest. So anyway, okay, we've already addressed the how specific do we need to know the pathways. Can you explain how to tell if redox reaction is inorganic or exergonic? We've talked about that. Can you go over the short answer of the practice exam? Oh man, I can, but I have to pull up the practice exam. So unfortunately, I have another computer sitting in front of me. So let me get to the practice exam here, and I'll pull it up, and then I'll state what the practice exam question is and go from there. I should have been prepared. I should have known that someone was going to ask that. Okay. Okay, yes. Okay, so our question in the practice exam is low blood glucose is detected by the pancreas that subsequently releases glucagon, a hormone that has widespread effects, including the activation of gluconeogenesis on the liver. Okay, given this information, predict the effects of gluconeogenesis activation on the following processes in the liver. rate of malate production in the mitochondria, glucose export out of the liver cell, and then predict the effects that a cytosolic malate inhibitor would have on the rate of glucose production in the liver and explain why. Okay. All right. So let's address this from a number of different ways, and let me go ahead and try and diagram this as well, if I can. Okay. I think I can do it. All right. So let me come back to this, and let's come here. And let's first of all just think about this from this standpoint. Gluconeogenesis now is going to be occurring in the cytosol of the cell, of the liver cell, because the extracellular signals from the rest of the body are telling the liver something needs to happen. Glucose needs to be replaced in the bloodstream. Okay, so that's kind of the big picture, is that the rest of the body is needing glucose. The liver is the thing that's going to supply that glucose in a time of fasted state. Okay, we didn't talk about glycogen breakdown, but one of the things that the liver will certainly do is break down glycogen in response to glucagon signaling as well. Okay, all right, so let me go ahead though and then just draw this. Let's consider this out here. Okay, and we're going to need two of these. Let's consider this essentially the cytosol of the cell, okay? And this is going to go all the way up to, sorry, not 2, but 1 glucose 6-phosphate. Okay? And this is going to then require, at the gap DH step, so I get my stoichiometry somewhat correct here. At that gap DH step, because gap DH is a cytosolic and a gluconeogenic enzyme, it's going to require that NADH be used. Okay, unlike glycolysis. In glycolysis, the NADH is made by gap DH. In this case, is used by gap DH. Okay, and so it's important that the body have some supply of this. Okay, now. We have then here, and this is an important factor here, and we'll try and do this in a couple of different ways, but we have here then this other compartment in the cell, okay, mitochondria, and we have to have pyruvate. Okay, now let's consider this, let's say like alanine, which is an amino acid that's found in our bodies, okay, in fairly high supply. Alanine is going to be our source of pyruvate. And all you have to do to make pyruvate from alanine is take an ammonia group off of it, okay? We won't talk about how that happens. We didn't really address that in class. But let's just say that amino acids, 18 of the 20 amino acids, by the way, can make glucose, okay? Well, let's just say this amino acid, alanine, can make pyruvate. And then what will happen here is this. Pyruvate will be moved into the matrix of the mitochondria. So we'll put out here cytosol. Okay? And we'll have now pyruvate in here. Okay, don't worry, I'm getting to the question in the exam, but we have to do this whole thing so we have some understanding of this, okay? All right, so then what will happen is that the first step in gluconeogenesis can occur, and that first step in gluconeogenesis is the production of oxaloacetate, okay? And the enzyme that does that is pyruvate carboxylase. Okay, and pyruvate carboxylase requires a source of carbon and also some ATP. So I'll just put ATP here. Okay, all right. And I'm going to leave some details out here, okay, because I want to get to the point of this, or the gist of this. Okay, now, here's where what we need to understand is that we need to resupply the cytosol with this, NADH, and the matrix of the mitochondria is a great source of NADH, but again, you can't get NADH out of the mitochondria as NADH because it has this membrane here, the inner membrane of the mitochondria, that simply will not let NADH out. Okay, and so let's go ahead and do this. Okay, let's look at this connection that this pathway is going to have with the malate aspartate shuttle, because now that oxaloacetate can become malate. Okay, and to do this, NADH is going to have to be used. Okay, and the enzyme that does this is malate dehydrogenase. And I'll just put DH, or this could be called MDH, malate dehydrogenase. Okay, that's the enzyme that does this. Now, going back to this, what I was saying, NADH, boink, can't get out, right? And I had a student tell me, they showed me in their notes that they had drawn something like this, because I've drawn something like this in my notes, that they've thought about the matrix of the mitochondria and that inner membrane as Captain America's shield, right? And that's a great way to think about it, right? I mean, Captain America's shield can block anything, including NADH moving out of the mitochondria. So anyway, okay, so haven't you missed this? I mean, I missed this. I haven't been able to teach you guys, so I missed interaction with you, even though this is a very strange interaction. But anyway, okay, so now malate can, Captain America's shield, this is the weakness for Captain America, okay? And that is that malate can go right through this, okay? And malate can get right through that membrane, and then it can, out here, it can be converted to oxaloacetate. Okay, and that will then take that NAD plus and make the NADH. Okay, now stoichiometry is off here, so don't get too hung up on the stoichiometry. Okay, but that's going to resupply our NADH. Now, I'm not going to draw the rest of the malate aspartate shuttle. Of course, we know that included in this would be movement of alpha ketoglutarate. So, for instance, we could draw an alpha ketoglutarate here, and it's going to go in, okay? And we could have the aspartate, and we could have the glutamate, and we could draw all that stuff that's going on here, okay? But I don't want to draw that because it's going to make this really busy, okay? Now, this enzyme, though, here, coming back to this whole thing, this enzyme here would be, again, MDH, same enzyme as what we saw in here. Okay, so MDH, right? So now, what's going to have to happen after this is that oxaloacetate is now going to be converted to PEP, and the enzyme that's going to do that is PEP-CK. Okay. My handwriting hasn't gotten any better because of the coronavirus. I'm sorry. You have to all forgive me. Okay. But that's PEP-CK. Okay. And then, of course, once PEP is produced, then we're going to start to make glucose. Okay. Now, coming back to our question. So let's look at this. Low blood glucose is detected by the pancreas that subsequently releases glucagon, a hormone that has widespread effects, including the activation of gluconeogenesis in the liver. Given this information, predict the effects of gluconeogenesis activation following the processes in the liver. Okay, so a rate of malate production in the mitochondria will go up or down. Well, look at what we're doing here. Okay, based off of this, we would say that the rate of malate production, if we start to increase this, then we have to increase this, and we're going to increase then the rate of malate production in the mitochondria. Okay, glucose export excretion out of liver cell. Now, something I didn't show you here, okay, is up here, because there's another compartment that this has to happen in. So this would be the ER. Okay, glucose goes into there and is converted to glucose, or glucose 6-phosphate goes into here, right? And then glucose comes out, and then it's going to leave the liver cell. Okay, so these signals will tell, of course, these liver cells to make glucose, and the glucose will be exported out of the liver cell. Okay, right. So the enzyme that does this dephosphorylation is glucose 6-phosphatase, and it's found in the ER. Okay, so it takes place in another portion of the cell. Okay, all right. Now, let me see here. Oh yes, coming back to this. So that was B. So that's going to increase. Glucose export out of the cell is going to increase. Okay. Predict the effects that a cytosolic malate dehydrogenase inhibitor would have on the rate of glucose production in the liver. Okay, well, what do you think here? Is it going to go up or is it going to go down? Start to think to yourself about that. But what's going to happen here is this. If there's a cytosolic malate dehydrogenase inhibitor, what would happen to the rate of malate of glucose production? Well, what we're saying is this can't happen, okay, because we've had some inhibitor there. Well, that can't happen, then you're not going to resupply this, or you're not going to resupply this, okay? And if you're not going to resupply, you're not going to resupply this, which means you can't make PEP. So if you can't make those two things, you're going to have a very difficult time making glucose. And so the rate will go down for that inhibitor, and the reason is because you're not going to make an ADH and you're not going to make oxaloacetate. Okay? All right. Awesome stuff. Isn't that awesome? Now, I heard a beep here. Do I have? Okay. Doing good on that. Okay? So, fun stuff. Okay. So hopefully that answers that question for you. Okay. Let's see here. On the practice exam, questions 56 through 60 are answered with the assumption that this does not take into account what happened in the prep phase. What does that mean? Oh, okay, so let's go ahead and look at that. 56. Okay, so the conversion of 2 moles of glyceraldehyde 3-phosphate to 2 moles of pyruvate by the payoff phase. Oh, so this is an old question because I used to use the terms payoff and prep phase. Payoff phase would be energy generation phase. Okay, in the glycolytic pathway results in a net formation. This does not take into account what's happened in the prep phase. Prep phase would be the energy investment phase. So in other words, you want to calculate the amount of NADH and ATP that would be made by the energy generation phase, not taking into account what's happened in the energy investment stage. Okay, so that's what that means. Okay, all right. So hopefully that makes sense to you. Okay. If not, let me know. Right. You can even unmute yourself and say, "I have no idea what you're talking about." Okay, and that would be fine. All right. How well do we have to memorize the different enzymes and intermediates? So that's going to come back to, I kind of addressed that. How specific do we need to, do we have to know the pathway? So honestly, let me come back to this. The fact that if you could do this, right, that's going to lead you a lot further along in terms of answering the types of questions that I would put on this exam than straight memorization of enzymes and intermediates. Okay, you have to be familiar enough with these pathways, the malate aspartate shuttle, the glycerol 3-phosphate shuttle, the lactate dehydrogenase, and also glycolysis and gluconeogenesis, to be able to diagram these things out in your mind, and then hopefully you'll be able to answer questions like this on the exam. So I am a strong proponent of not only just diagramming the pathways, okay, and you can diagram them in their nitty-gritty detail if you really want to. Name the enzymes, name all the coenzymes, name all the products, all that kind of stuff, and that would be great, okay. But I would also want you to be able to diagram them in connection to each other and also in the compartments of the cell, kind of like what you see here. There are 3 different compartments that are being used, the ER and the cytosol and the matrix of the mitochondria, okay, and how they interact with one another, and then how, like, if I start to disrupt this portion of one pathway, what will that do to the other pathway? Because we want to have an integrated view of metabolism, okay? Okay, so hopefully that at least leads you in the right direction on how to do this, okay? Do I have to be able to do math? No, so there's not going to be any question where it requires you to do math. So just like the first 2 exams, I would expect you to understand the implications or the meanings of equations, but not. I'm not going to ask you calculate, right, the delta G given C1, C2, and the delta psi of a particular membrane or something like that. So nothing like that. Okay, but I would expect you to know that, okay, well, if C1 is bigger than C2, then it's not an electrochemical gradient, then the movement of the molecule will be exergonic. Or, right, if C1 is greater than C2, but we have an anion moving into a negatively charged compartment, well, then maybe it's not exergonic because now we have a favorable concentration gradient, but we have an unfavorable electrical gradient, all right? And therefore, it's hard to say if it's exergonic or not, okay? You'd have to actually do the math, and I'm not going to have you do that, but you couldn't say that that's always, the scenario that I just stated is always exergonic, because we have an anion moving in a negatively charged compartment, okay? All right, so hopefully that helps you out with that. Okay.
Excuse me, Dr. Solberg?
Yes.
Will we be given the equations on the exam?
Yes. Yes,
Okay.
You will be given the equation. So on Canvas, if you look at like your practice exams, which of course just a Word document, you see above them you have abbreviations and some potentially helpful, useful information right above the matching questions. On the very first matching question on Canvas, I put all that helpful information and the abbreviations and everything. Okay, so it'll be at the top of your exam in the first matching question, so just refer back to that. So I'm glad you asked me that. I definitely will supply those things. The other thing that I want to go ahead and address, let's just address this right now, because while I'm thinking about it, you can use scratch paper on the exam. So you'll have the ability to use 2 pieces of scratch paper on the exam, because I know people like to draw things out and use their pen and that kind of stuff. So go ahead and do that. You won't be able to take like a bathroom break or something like that. So if you get up and you leave your exam, you're going to be tagged. I'm sorry to say this, but you're going to be tagged, and it's going to generate a report and all that kind of stuff. So if there's anyone else in the room with you, that's going to be tagged as a problem too. I just gave an exam this Wednesday with about 100 people in the class. It went off really well, although there were some students that right after the exam wrote me and said, "Oh, my brother walked in during the exam," and and that kind of stuff. So those kind of things I know are going to happen, but please let me know about that. That way I have an understanding when I get this report saying that this happened, then I'll have an understanding of why that happened, okay? I really do want to take the mode that you guys are trustworthy, you're not trying to cheat. So that's the mode that I'm going to take with this exam, so, okay? All right, so good questions. I'm glad that was asked, okay? And feel free to interrupt me at any time. Just unmute yourself and interrupt me, okay? All right. Are the glycolysis, gluconeogenesis, malate, aspartate shuttle, glycerol 3-phosphate shuttle, lactate fermentation the major concepts that will be covered in the exam? So don't forget about lecture 9 and 10. So there's about equal amount of material from lecture 9 and 10 as there is from lecture 11. So while there's a lot of information in lecture 11 because there's a lot of detailed information, there's still a lot of information in 9 and 10, right? So if you look at, again, the learning objectives, I walked my way through those learning objectives and caught learning objectives from 9, 10, and 11, okay? And with chapter 10, we got steady states versus chemical equilibrium. We've got, metabolic regulation, which is a major topic in biology and biochemistry. So, so those are all things that you're going to want to keep in mind as you move forward. Okay. So there's not the major things. They are certainly covered on the exam, though. We have to be able to do math. I've already addressed that. Malate aspartate shuttle and why we need forward and reverse direction. Okay, so that's a great question here. So let me come back here. So in this case, this is the malate aspartate shuttle running in the reverse of what we've normally seen. Okay, because or not what we normally saw, but when I first presented to you the malate aspartate shuttle, I started here. Let me come here, kind of continue this on from what we were doing above. I started with something like this. Glucose is converted to 2-pyruvate. Okay, and 10. Sorry. What's going on? Why is it not letting me erase? Okay, I'm getting some poor performance here. I hope I don't lose this. There we go. NAD plus. There we go. It's back to NADH. Okay, so that's the glycolytic pathway. Okay, and then we have to, of course, resupply NAD plus with the glycolytic pathway if it's the pathway that's running. And so what I did initially was I presented it this way, okay, and that is that this oxoacetate, okay, can be converted. Actually, I did it in here. Okay, my eraser is not, so just ignore that. My eraser is not really working, but oxoacetate was converted to malate. All right, and this NADH was used to do that. Okay, and that's how NAD plus was resupplied. And then malate, of course, went into here. Okay, malate, and it was converted to oxaloacetate. Again, I'm sorry for my bad handwriting. And this is the NAD plus, goes to NADH, right? And I'm not going to again draw all the gory details. You can look at that on your own time. Okay, but notice that this is going in this direction. Okay, and we'll continue on in this direction if I was to draw the rest of it. Okay, when in this case, okay, it's really going, for gluconeogenesis, is going in the opposite direction. Okay, it's coming out of the mitochondria first and then back into it. Okay, and it is taking, it's resupplying NAD plus in the cytosol rather than, I'm not NAD plus, but NADH rather than NAD plus. Okay, so you have to keep that in mind, that it can run in two different directions, and every enzyme found in the malate aspartate shuttle is reversible. Okay. All right, hopefully that's clear. Okay, there we go. Can we go over major diagrams and processes? So let me put that one on hold, because I want to get to some of these other questions, because that might drain the rest of our time. Okay. The major diagrams and processes would be the malate aspartate shuttle, and glycolysis and gluconeogenesis and lactate fermentation, and the glycerol 3-phosphate shuttle. Those would be like big ones. And then in chapter 9 there would be the movement of glucose into the membrane. That would be a big one. And then in chapter 11, at the end of it, we talked about glucagon and insulin signaling. Okay, so there's a lot of information there. There's a big diagram. Okay, but if I were to try and go over every single one of those, I wouldn't have time. Okay, so I would ask that I've posted annotated notes. I would ask that you look at those annotated notes, that you go back to those portions of those lectures and listen to them, because that's really going to be the best way to do that. Okay. Now, can you explain and provide an example of covalent modification? Yes, I can. Covalent modification is essentially just like it says, the covalent attachment of something to a particular enzyme. Okay. And so we actually gave an example in class. Let me go ahead and do this. But there was this enzyme, remember, the bifunctional enzyme. Okay, I could give lots of examples, because covalent modification is there's a lot of covalent modification that occurs to regulate pathways, but I'm going to go ahead and use one that you've already seen, so that I'm not introducing like a whole bunch of new material to you. Okay, but we had that bifunctional enzyme PFK2, remember, and FBPase2. Okay, that can undergo what would be called covalent modification so that it would become this. There would be this covalent attachment of, let me go ahead, I'm going to go ahead and actually put it over here so it's out of the way of things. But it'd be a covalent attachment of this group, a phosphate group. It's always been drawn in class as just this P with a ring around it, but that will be the functional group, or an organic chemistry functional group that's been attached to the enzyme. Okay, and to do that, you have to have an enzyme that we'll just call it pKa. Okay, so it can go by other names. CAPK is another one. Remember, if you go back to that lecture, as pKa, aka CAPK. Okay, right? So if you remember that, I try to make jokes out of these things so that you can laugh and try and remember things as well. But pKa is protein kinase A, and it will take the phosphate off of the ATP. Okay, and take one of those phosphates off of it and attach it to the enzyme. So it's coming off of ATP and making ADP, of course, and it's being put there. And there's this entire enzyme that needs to do that. And when that happens, then again, we get sunbeams coming out of this, and that becomes active. Okay, and then in this case, because it's become active, then it's going to break down. The FBPase2 break down fructose 2,6-bisphosphate, making fructose 6-phosphate. Okay, but that's only active because it's been phosphorylated. Okay, you can dephosphorylate this going this way, take the phosphate off. Okay, take the phosphate off, which is, this is, we just call that PI. Okay. You have the phosphatase to do that, and then in this case, then what becomes active is PFK2, which will then make the fructose 2,6-bisphosphate. Okay, but that's an example of covalent modification. It's regulating this enzyme. Okay, in this case, it's regulating an enzyme that's responsible for regulating the breakdown or the production of F26BP, which then F26BP is an allosteric regulator of glycolysis and gluconeogenesis. So when we start to look at regulation of metabolism, it's always going to be tiered. Okay, there's always these tiers to it, meaning not always, I guess, but usually tiered. There can be a covalent modification that causes a change in the production of an allosteric modulator, and they will allosterically modulate something. Okay, it can be really complicated really fast. Okay. Can you explain and provide an example of, okay, I already did that. How much physics concepts will be in this exam? Like how much of the thermodynamics will be included? So we covered a lot of the thermodynamics in chapter 1, like the major differences between delta G and delta G prime naught. So I'm not going to be recovering that or rehashing a lot of that. But you know, whatever we talked about directly in chapter 10, thermodynamic concepts will be something that we, of course, address on the exam as well. Okay, big concepts. Like a big one would be the difference between a steady state and chemical equilibrium. That's a really important concept to try and understand. The first law of thermodynamics and how energy is being transferred is going to be a really important one as well. Right, so we looked at how you can have nucleophilic substitution reactions that are important in transferring energy. We talked about redox reactions as being important in understanding how those transfer energy. Okay, you know, in the case of nucleophilic substitution, we could have substrate level phosphorylation of ADP making ATP from something like 1,3-bisphosphoglycerate. Okay, so those are fairly important concepts in this. Okay, so review those, right, and look at the slides of chapter 10. and make sure that you are understanding those concepts. Now, again, I'm going to point you to learning objectives, because learning objectives really point you to the type of thermodynamic questions that I would ask. Okay, and I don't have those pulled up right now, but you should be able to pull those up fairly easily and look at those. Okay, so make sure you look at those. Okay. The malate aspartate shuttle. Okay, I think that we've covered the malate aspartate shuttle. We've gone over it quite a bit in connection to glucose and gluconeogenesis and glycolysis. So I'm going to leave that one as it is. Do we need to know what the chemical structures are of the molecules and what is added or deleted in each individual reaction? Oh, absolutely not. So that would be way beyond what what I would expect you on this one. So no, you don't need to do that. How much time do we get? Is it more than 50 minutes because I'm a really slow typer? So it is. So that question has been addressed in the announcements of Canvas, and I'm just going to pull that out right now. You really need to look at. There has been 3 announcements about the exam. And Proctor, how it's going to be Proctor and all that kind of stuff. Please read that, and please read those so that you are really well prepared for the exam on Monday. So, but you do, in those announcements, it said that you would actually get 80 minutes. So I'm giving you 30 minutes longer because of things like you're a slow typer or possibly an internet lag time or something like that. There's lots of things that can go on when we are doing an exam online versus just face-to-face with a regular old pencil and eraser. Okay, so 80 minutes is what you're going to get. You're going to have a 10-hour block of time where you can log on and take the exam on Monday. So not everyone's going to be hitting the servers hopefully at noon on Monday. Okay, you can take it at noon on Monday, but you could take it at 8 a.m. on Monday as well, or you can take it at the exam closes at 6. Okay, so I am telling people, please do not start your exam later than 4 o'clock. Okay, that way if you run into problems or whatever, you won't have an exam auto submitted halfway through. Okay, because it will auto submit at 6 o'clock. Okay, so keep that in mind. Okay, and those are all, of course, Mountain Time. Okay. What will be the most important pathways to memorize? I haven't talked about. Like I said, I haven't asked you to memorize any pathway, so I would expect you to be familiar with all the pathways that we've talked about. Okay, to the level that I've talked about them in this review. Okay, can you explain allosteric regulation and what negative and positive modulators do? An example of that. Okay, so allosteric modulation is very similar to, and by the way, I'm going to run this until 1 o'clock. So it's almost 12:50, which is normally when we meet in the class. I'm going to go ahead and run it until 1 o'clock, okay? So we got a little bit more time to get through some of these questions. All right, but allosteric regulation is the same thing that we talked about with hemoglobin, and the fact that you can have negative allosteric regulation or positive allosteric regulation, okay? So in the case of enzymes, we have either an increase in specificity for a substrate or a decrease in specificity for a substrate due to some allosteric effector, okay, or allosteric modulator, okay, meaning that we have, let me go ahead and draw an example of this, meaning this, we have some enzyme, right? Let's say we have enzyme, just call that enzyme, and let's just say that it's right, it has some level of activity, but it's essentially not very active. Okay, so we'll just put a little minus sign in it. Okay, that can be bound by some small molecule. Okay, a modulator. Okay, so let's call that a modulator. All right, and that can, let's say, cause a change in the conformation of the enzyme, because that's what modulators do. So it's gone from a square to a circle, and in fact, it's gained mass, apparently, because it's a bigger circle than it was a square, okay? So hopefully you're laughing at that stupid drawing. But anyway, okay, so we have now the modulator bound to this, and now we can say, oh, this is now active, okay? And so this would be a positive modulator, okay? Now, it's bound at a site, because it's a modulator and it's an allosteric enzyme, it's bound at a site that's not the active site of the enzyme. Okay, and what this could do So if we were to draw this graphically, okay, let's just say that this is our enzyme without a modulator. Okay, that's no modulator. Okay, and then with the modulator, it could go like something like this. Okay, so this is positive modulator. And in this case, it's had an increase in specificity, which means a decrease in K-half. Right, so we don't use the word Km because these are not hyperbolic enzymes, they're sigmoidal. Right, so we would say that the K-half for this has increased. Okay, and we measure a K-half like right here. Okay, so we have K-half here, K-half here. Okay, and this would be 50% Vmax. Okay, and so then our K-halves would be this, and smaller K-half would mean greater specificity. Okay, and that could be the case because of positive modulation. Negative modulation, the curve would shift in the opposite direction. You can also have a positive or negative modulator that will change Vmax. Okay, so in other words, it would look something like this. Okay, VO substrate concentration, no modulation, and it has some Vmax that it's approaching here. Okay, in the case of positive modulation, it could look something like this. Okay, where we have a new Vmax. Sorry, my drawing is really bad. Okay. Now we have a Vmax 2 here, and this would be due to positive modulation. Okay. So in this case, our Vmax has increased, and the reason that a Vmax would increase is because the K-cap's gotten higher, okay, or larger. Okay. All right. So hopefully that answered that question. Can you talk about the roles tryptophan and tyrosine side chains play in the membrane? Okay, yeah, so that's going back to chapter 9. So in chapter 9, I didn't spend a lot of time on this, but I don't want to draw something really crazy here, but tyrosine and tryptophan, if you go back and look at those side chains, they are hydrophilic and hydrophobic, meaning they're amphipathic. Okay, like tyrosine, for instance, let me just draw this because I know that not everyone remembers the structure of tyrosine. Okay, but tyrosine side chain looks like this. And then we have the alpha carbon, right, and the rest of the amino acid. This is the side chain of tyrosine. Well, that's amphipathic because it's got the hydroxyl group that's polar, and then it's got the benzene ring that's nonpolar. Okay, and so these, and tryptophan has a similar type of amphipathic nature. So these play really important roles right at the interface between the polar head groups of the lipids in the leaflets of the membrane and the hydrophobic tails. So this, for instance, could be contacting the tails, whereas this could be contacting the polar heads. And so you'll find them enriched in the integral membrane proteins right at that interface of the inner and the outer leaflets of the membrane, right? So if we have our membrane here, let's say, and then we have our membrane protein here, okay, then you would find right here in the structure of the protein, not always, but you'd find an enrichment of tyrosine and tryptophans because they can contact the hydrophobic tails. Okay, and of course all of this would be hydrophobic. And then we've got the polar head groups would be right here. Right, and the tails would be going something like this. Okay. And so tyrosine and tryptophan will be found like right here, contacting the hydroxyl group, contacting the polar head group, and the benzene ring contacting the tails. Okay. Right. So they play an important role in like anchoring the membrane proteins to the membrane. Okay. How does ProctorU operate? Again, announcements. Look at those announcements. So essentially, you will have, if you actually look at this, come up here because you can see this. This is my browser. No, that's not it. Of course, this is the one browser that I don't think I have added it to. Okay, so you can't see it here because I haven't activated. I haven't done the Proctor. I haven't done the ProctorU extension onto this browser. Okay, but if I had, then up here you would see this little owl icon, and you could click on that, and it might say log in, and then you have to generate an account and go ahead and do that. Or it might say, log out, and it says log out, then you've already made an account and you're ready to take the exam. So and then once you go on to Canvas, then you'll click on the exam to take the Canvas, and as long as you've logged into the ProctorU, then Canvas will automatically, ProctorU extension will automatically populate a password to Canvas that will then allow the exam to open to you, and then the clock will start essentially, and then you can start to take the exam. Now, when that happens, your exam is going to go full screen, and it's going to remain full screen until you submit the exam. Okay, so that you can't use anything else on your computer. After you submit the exam, just go ahead and hit F11, and it'll take you out of that. Okay. Our channel is always passive. Yes. Will you post this recording? Yes, I will post this recording. Could you please briefly go over Michaelis-Menten for Chapter 9? Briefly, yes. In fact, this will probably be the last question for our little recording here. Okay. All right. So the Michaelis-Menten stuff for Chapter 9 is the exact same as it was for Chapter 7. Exactly the same. Okay. It's just that we're applying it to transporters. Okay. So, but when we think about it here, Michaelis-Menten, right? We have our substrate, but in this case, we'll just say that the substrate is on the outside of the membrane. It's going to be moved to the inside of the membrane. That's all that's happening. Okay, is this the movement of this, and we can plot some rate versus the concentration of the substrate on the outside. As the concentration of the substrate on the outside goes up, then the rate of the transporter goes up until it reaches this maximal velocity, at which point we say it's saturated. Okay, and it's saturated for the same reasons that enzymes saturate. There's just so much substrate that the only thing that's slowing the movement of the substrate across the membrane is the rate-limiting step of the mechanism to transport the substrate across the membrane. Now remember, for carriers, they have those different steps. Okay, they have the recognition step, right, and then they go through that whole cycle that we talked about. Okay, all the way till you get the translocation and the reset step. Okay, so it's, I think it's recognition, translocation, release, and then reset. Okay, those are the steps. I think those are the steps that are found in the transporter, and one of those can be the slowest step. And because of that, then you will have at some substrate concentration when it's saturated, then you'll have some rate limiting step. The rate limiting step for that, okay, gets a rate constant. We call that rate constant K-cat, just like we did for enzymes. Okay, and then you'll have a Km, and the Km is a measure of specificity of the transporter for its substrate. Okay, and when you think about Km and specificity of the membrane or of the transporter, then that is related to the selective permeability of that membrane. Okay, so for instance, if you have a transporter that has a high specificity for a particular substrate, then that membrane will be permeable to that substrate, meaning that it will allow it to go through. Okay, if it doesn't, then it will be impermeable to that particular substrate. Okay, so let's go ahead and end there. I think that it's 1 o'clock. I'm going to let you all go your separate ways. I will go ahead and let me come back to this environment here. Come back here. I will go ahead and see if there's anything in the chat. Okay, so no one's typing anything in the chat. Okay, good. I'm going to go ahead and post this recording. All right, and so if you did have significant lag issues, then you should see that posted by the end of this afternoon. Okay, have a good weekend, and good luck in the exam on Monday. I will be sitting at my computer all day Monday. I'll be responding to emails. I will be giving priority to you guys and of course the needs that you have, especially if you have problems with ProctorU. I hope that everything's going to go really well. It went really well for my last class. I only had 2 students that had problems. Okay, so I'm seeing a lot of people say thank you. You're welcome, absolutely. And I think you're all great. I miss you. So I will see you though, or interact with you, I guess, in some other way. Probably do the same thing for exam 4. Okay. All right. Take care. Bye.